We certainly don't have time for a wordy introduction because Rich has probably way more than 30 minutes, but you're looking at one of the world's great authorities in aging everything. And it's been a pleasure to work with Rich for getting on towards 10 years. He helped us escape from the inbred diagonal of using just BXDs. So we've been collaborating with Rich and the whole team at National Institute on Aging using the so-called Het3 mice, which in none of Rich's papers does it explain what that acronym means, but I asked him, and stands for University of Michigan Heterogeneous Number 3 of whatever Rich and colleagues tried back in the late 90s and early 2000s. More or less, right?
Yeah, that's exactly right.
But the rest of the world is left to guess what Het3 means. But I think Rich is just too smart for his own good because he assumes people will figure it out. So I figured it out, but with some help. And from my point of view, I misintroduced him on an email recently as a card-carrying pathologist, which he says, nope, Rob, totally wrong. If you look at his biosketch, he's actually got a background in genetics, which kind of explains.
I got the first ever PhD in human genetics at Yale.
Wow.
Actually, because they had a brand new department of human genetics, and my boss became one of the founding members, and I just slipped in before the door closed. So.
That's a great story. I did a postdoc right next to the Department of Genetics and knew most of Ken Kidd. He probably wasn't there then.
Yes, he was.
Was he?
He was a baby, but he was there.
Okay. Anyway, so Rich, if there's any other introduction you want to give about yourself?
No, I just wanted to talk about the science. So a lot of people like to do transcriptome research. They figure once they've got the RNAs, they've done something. And so the point of my talk today is no, you've missed everything. If all you have is transcription. Tomorrow's talk is entirely different. Tomorrow's talk is about drugs that slow aging in mice and using them to gather information about pathways that can be used to slow aging in mice and how aging is controlled and how diseases are related to aging. Today is two vignettes that don't fit into that rubric. The goal here is to look at two nice stories coming from junior faculty members in my group. One is on control of proteome by selective mRNA translation, and the other is control of proteome by selective protein degradation. There's evidence now that an awful lot of the proteomic change is not transcription based, but is based upon degradation and translation. That's what we're going to talk about. So tomorrow we'll talk about the ITP and aging rate indicators. So here are the things you need to know just to sort of get oriented. We work with lots of different kinds of slow aging mice. There are 5 mutants. Snell-dwarf and Amesdorf have growth hormone problems. Abbé has problems with IGF-1 binding proteins. P10 is a famous tumor suppressor and growth hormone receptor knockout. They all live 20 to 40% longer than their littermate controls. We have 4 drugs that we use a lot. Rapamycin, which increases lifespan by 26%. Acarbose, which works in both males and females, but a lot more in males. And then 2 others that are male specific. And that's important because some of these changes turn out to be sex specific. So let's talk about cap independent translation. The first, yeah.
Is there a reason we have so many ones that are specific to mice?
I wish I knew. Let's talk about that later. I don't know the answer, and I really wish I knew. So this was the first observation that sort of got Gonzalo onto this. He was looking at 2 proteins of special interest for his work, NDRG1 and MGMT. They form a complex that helps take care of DNA repair. And he looked at the DW mice, that's the Snell-dwarf mice, the long-lived ones, and by gosh, both proteins were up in liver, muscle, and kidney. And so he said, yeah, it's probably just trend. It's also true for the growth hormone receptor knockout mice. Both proteins were up in all 3 tissues. But surprisingly, when you looked at the mRNA, there was no change in the mRNA. The proteins were up 3-fold, but none of the mRNA. So why could that be? And then he said, well, look, let's try a drug. Let's try rapamycin. Let's give it in vitro. That's on the left where you're hitting cells with rapamycin, or on the right, you're giving mice rapamycin. The cell thing is 48 hours, the mouse thing is 7 days, and the proteins went up, and the mRNA did not change. So what's going on is this, most mRNAs, that's the thing going from left to right there, have a cap over on the left, the M7G PPP cap, and that's where the 40S subunit binds, and then it sneaks down to the start site and starts making a protein. But you can block that. A lot of bad things can happen to cells that block that, and then a special subset of messenger RNAs just keep puttering along. They're special because in the 5' untranslated region, they have 6 methylated adenosine residues. And the 40S ribosomal subunit can bind to that and then start moving down till it hits the translation start site, and it can make an mRNA, even though the cap-dependent translation has been stopped. It makes cap-independent proteins.
At a much slower rate.
No. But selective. Very few mRNAs can do that.
Okay.
The things that influence this, there are 2 writers that put that M6A site, they're called metal 3 and metal 14. And there are 2 readers, or at least 2, YTHDF1 and YTHDF2. They had been known to be involved in mRNA stability, mRNA translation, and it turns out one of the ways they do that is by seeking out 6 methyl adenosine sites in the 5' untranslated region. So Gonzalo said, look, what happens if you give him a drug that blocks cap-dependent translation? And the only thing left is cap-independent translation. This is fibroblasts, and the protein data are shown over on the left there. MGMT goes up, NDRG1, and it turns out 4 other things shown there: PGC1 alpha, TFAM, which is a mitochondrial biogenesis factor, LONP1, which helps protect mitochondria against oxidative injury, and surprisingly, YTHDF1. This is a positive feedback loop. That's the reader of the M6A signals. That goes up too when you block cap-dependent translation. The stuff on the right is controls, and since we don't have much time, I won't go through all those. But it's cap-independent proteins that go up, not too surprisingly, when you block cap-dependent translation. Then he did it again by giving the same drug to mice, and in the mice, you get exactly the same thing. All those proteins go up, including YTHDF1 in the mouse liver if you block cap-dependent translation. And when you take fibroblasts and you block cap-dependent translation and you're stuck with cap-independent translation, the cells become resistant to DNA damage, resistant to ER stress. We really need to track this down and look at other forms of stress resistance. Okay, so we've got,
Animals with cap-dependent translation block, they become more resistant to the.
that's what 4EG1, EGI1 does. Yes, it blocks cap-dependent translation and thereby encourages cap-independent translation. That led to the stress
And
resistance.
it increases the resistance to, to toxic effects.
Yes. Now we want to do this again, and we want to do it with a wider range of stresses, but that's the published data. Okay, so we've got one drug, rapamycin, and I've shown you only 2 kinds of mutants, so there are more. What about other drugs? Is this a common unifying factor that extends across many different kinds of slow-aging mice? What about acarbose, which is lifespan beneficial for males and females? Or 17-alpha-estradiol, which is lifespan beneficial only for male mice. So this is the acarbose slide, and the proteins we're looking at here are MGMT, TFAM, and NDRG1, top to bottom, males and females. If you look at the first 2 bars, that's young and old. Young is black, old is white. So aging, old in this case is 22 months of age. Aging leads to a decline in all of these cap-independent protein targets, but not if the mice have had acarbose. The 3rd bar in there got acarbose 16 months before it was killed, and the bar all the way on the right got acarbose 4 months before it was killed. That is from only 18 to 22 months of age, and it doesn't matter. In both sexes, the age-associated decline in production of cap-independent proteins has been blunted by these anti-aging drug, acarbose. No change in mRNA levels. This is a translation control story. And this is a control. These are the cap-dependent targets. Acarbose does not block all translation. It blocks aging does not block all translation. It blocks translation of the cap-independent proteins, and the drugs don't have any effect on that. Not too surprising.
Just a little more on cap independent. How many proteins?
I wish I knew.
Oh, that was the question that.
No, that's a
he asked.
different thing. I wish I knew. Which is why.
That's the same answer. though.
Why? No, yeah. It's a really good question.
Are they like stuck if an mRNA is cap dependent? Is there like a cap dependent mechanism, or are they just stuck to the gap?
The cap independent proteins are the ones that are fortunate enough to have
Okay.
M6A in the 5' untranslated region.
Okay.
And they've got to be able to, I'll show you and they've got to have YTHDF1, which turns out to be elevated in all the slow aging mice. Okay, and so what about, now let's move on to 17 alpha estradiol. This is the one that has the lifespan benefit in males only. So the males are shown on the left there, and not surprisingly, because it's the exact same data, aging still leads to a diminution in MGMT, TFAM, and DRG1. But in the males, just like acarbose, 17-alpha estradiol causes a blunting. Actually, they go above the level that you see in young mice. There's more of those proteins in the drug-treated males. But in the females, the story is quite different. For the bottom two proteins, it just doesn't work in females. For the top, it works a little bit, doesn't push them above the level of young. It does push them above the level of old, but only if they've been on it for 16 months. So there's a clear sex dimorphism, which parallels the sex dimorphism in the lifespan benefit. Okay, I'm going to show you one slide from tomorrow's talk just to sort of provide a link, but it's a really interesting one. There's a protein called GPLD1. I'll talk about it a lot tomorrow. One of the reasons it's interesting is if you give it to mice, they retain cognition for a long time. That's good. So it's good for thinking. Another reason it's interesting is that if you exercise a lot, if you're a mouse or a person, it goes up. That's probably why exercise is good for thinking, GPLD1. So that's why we're interested in it. That's a lot of that is tomorrow. So here's the thing I wanted to talk about today. Those two mutant mice have a lot of GPLD1. They have it in the liver, males and females, and they have it in the plasma, males and females. That may be why they're so smart when they get old. But surprise, gosh, it's not transcriptional control. There's no change in GPLD1 messenger RNAs at all in these slow aging mice. So what happens if you take a transgenic mouse that has a lot of YTHDF1? We've already shown elsewhere that they turn on an awful lot of cap-independent proteins. YTHDF1 is the reader. It reads that M6A signal and it switches the whole mouse to cap-independent protein production. And voila, GPLD1 turns out to be a cap-independent protein, something you would have missed completely if you were stuck in the old-fashioned world of transcriptional control. And you can see on the right there, not a change in mRNA levels. So what if you take some cells and you dump on 4-EGI1 to block cap-dependent translation? Well, there's no change in the mRNA level, that's over on the right, but the protein goes way up. So either in a cell biology setting or in a short-term mouse treatment study, you can turn on this terrific protein. By translational control, it's not controlled at the transcriptional level. You'll hear a lot more about GPLD1. So this is the last slide in my cap-independent vignette. This is what we're thinking of doing. We are, to satisfy Rob's needs, building a larger list of cap-independent translated proteins by comparing 4-EGI1 treated mice to normal mice, for instance, or YTHDF1 mice to normal mice. We're checking in a lot of other mutant and drug-treated mice, and the answer is yes, CID proteins go up in a lot of other mutant and drug-treated mice. Once we get chunks of muscle and fat from old people, we're going to check cap-independent proteins in humans to see if the fittest 60-year-old people have a lot more of them. And if we ever have collaborators interested in mapping QTLs, we could possibly map QTLs that control cap independent translation. They'll never get there by looking at mRNA levels. They have to look at the proteins. We want to do some molecular biology by connecting it to YTHDF1 and metal-3. And it's a different story, which I can't talk about. There's another protein called C0, which is also changed in slow-aging mice and which interacts with YTHDF1. We certainly want to screen lots of drugs to find drugs that turn on cap-independent translation. And once we have some, we're going to give them to mice and they're going to live forever because they will have cap-independent proteins going up just
This
as.
is why you're not optimistic about that.
Pardon me?
Okay.
Yeah,
I was wondering
that's
you're
why I
not
really want to
optimistic.
find drugs that turn on the cap-independent proteins.
If you block the cap-dependent proteins in
Right.
the mouse globally or in just.
Yeah, rapamycin does that.
Hmm.
Rapamycin does that.
How long do, so you can basically shut down, or do you just turn it down?
Turn it down. If you shut it down completely, you have a dead mouse.
That's what that was,
Right.
yes.
Yeah.
That's where I was going.
Yeah,
Uh-huh.
you shut it down some,
Okay,
and that allows the cap-independent proteins to come back.
so when you shut it down, you're shutting it down by a factor of,
I don't know. Good question.
But but if you just turn up the CIT,
Yeah,
that also,
we'd like to have drugs that do that.
yeah.
So far, the two drugs that we have that do that, do it by turning down cap-dependent. So what we want to do is do things that like turn on YTHDF1 or turn on metal-3 and metal-14. And can therefore increase, we hope, the cap-independent protein set without messing with cap-dependent translation.
Do you know enough about the cap-independent translated proteins across species to see if there's conservation of what?
Yeah, that's a really good idea. Greg Tomline, who works with Vera Gurbanova at Rochester, we have given him liver, heart, kidney, and plasma from 60 species of birds and rodents and primates. And I got the list of proteins he's evaluated yesterday. Okay.
yeah, cool.
Now, the hypothesis that is going to work both within species and across species, which is intuitively very attractive, is probably wrong. For instance, GPLD1, which I've shown you, is elevated in all the slow-aging mice, or some of them. I'll show you all of them tomorrow. That turns out to go down, at least in plasma, in the longer-lived species. The opposite of what we expected. I was very upset when Freddie showed us that data that it's airtight. He's got already in 12 species that he's done. He's looking at all 60 of them, but the first 12, you get a nice straight line and it points in the wrong direction. So
Same with body size. In size,
that's a very complicated, it's not a complicated issue, but it's a different issue. It's a very,
yeah, but also different, interspecies.
yes, but that's because of two entirely different factors. Interspecies is
Yeah,
IGF-1 growth hormone.
Yeah.
In across species, it's joint selection, large
Because
body
are
size, and low hazard niches. Not the same molecular thing at all.
Can you go back one slide? Now, why do you want to check if the healthiest subset of 60-year-old people?
I bet
Because you're
that
already
the healthiest subset of people have high levels of aging rate indicators. I bet that they are aging slowly.
Yes, but what are you comparing it against if you're only checking the healthiest subset?
I'm checking against all the other people that didn't get into the healthiest subset.
Okay, fine. Okay, thank you.
It is a good question. I've asked the variant of that to Rich, because as part of the application we're putting in, he wants to look at 6-month mice. And said, well, why don't you look at 12-month or 18-month or 24-month?
Because most of our data comes from healthy young adults, and the hypothesis is that if you're aging slowly, you're going to have slow aging rate indicators. But this is tomorrow's talk. So let me show you one thing that I'm hoping will shake your world. It's not my data, it's published data. It's from Gary Churchill, Ron Corstanja, and a batch of other people at the Jackson Labs. What they did was they took an inbred mouse, of all things, black 6, and they measured the proteome of black 6 mice at multiple ages, 6, 12, 18, 24 months. This is kidney data, but they've also published another organ that shows the same thing. They published heart data also. And they've calculated for each protein, how much does it change with age? And then with the same tissue chunks, they measured the RNA for all of those proteins and asked for the RNA, how much does that RNA change with age? And then they compared the RNA score, which is the horizontal axis, to the protein Z score, which is the vertical axis. So the blue dots are those proteins which went up with age, and so did the mRNA. The green dots are the ones where the mRNA went down with age, but the protein went up with age. The red dots are the ones where the mRNA went up with age, but the proteins went down with age, the wrong direction. And the surprise is that the R value, not R squared, but R, was 0.24. So R squared is 6%. That is 6% of the age effect on protein levels in black 6 in kidney can be attributed to corresponding underlying changes with age in mRNA. Most people intuitively, if they've never looked at the data, would guess that would be 90%, because that's why they're wasting their time looking at the transcriptome, why they're spending all their time looking at which parts of the chromosome are crunched up or hidden somewhere, or are they doing attack-seek stuff, or looking at epigenetic changes, because they think transcription is the end game.
Hey, but we
That's
have a
wrong.
hammer and it's called the transcriptome hammer.
That's
Don't
right.
disabuse it.
That's right, that's right, that's the problem. So that's the implication of this.
People know the difference.
Not everybody. I think
Too
we
few,
do.
everyone in this room
See the
does
thing.
now. Yeah, it's largely, it's been appreciated that there's not 100% correlation. But what I found shocking, and which people are just gradually incorporating into their worldview, is that it's not just different from 1, it's not that much different from 0. It's really low.
If we figured out a way to select the cap independent mRNA,
Yes,
like using some kind of hybridization.
right.
I wonder if that would have a better correlation with the H1 genes.
I'd like to ask that question. That's a good
I mean,
one.
that would be.
I myself am not that interested in does the thing change with age. I'm really interested in does the thing discriminate slow aging from normal mice? But
Right.
whether it changes with age is a good problem for those people who are interested in things that change with age. Now, here's the problem. And this is for you. It's 4:24. I have another talk. It's about 20 minutes, just like this one. I'd be eager to give it. I don't want to waste
Go for
people's
it.
time.
Go for
Okay.
We'll
So-
hold our questions.
Here are the take-home messages. Slow aging mice may mold the proteome by selective translation. And if all you have is transcriptome data, you are missing most of the action. We're going to turn to a separate issue, though it's related to proteome molding by non-transcriptional means. It's Joe Endicott's work. It's on chaperone-mediated autophagy. Most people think of autophagy as the blue thing on the left there. It's macro-autophagy. There are vesicles that form around organelles and stuff and dump into the lysosome for degradation. We're not going to talk about that. Other people think about micro-autophagy, the green thing at the lower right, where the lysosome gobbles bits of cytoplasm and whatever's inside. Not going to talk about that. We're talking about the stuff that's at the upper right-hand corner. It's chaperone-mediated autophagy. It's selective for specific proteins. The thing they have in common usually is a sequence KFERQ, but there's some variation in that. I'll leave that aside. If you've got the right kind of sequence, you bind to a chaperone, HSPA8, and with the help of LAMP2A, then and only then do you get moved into the lysosome for degradation. Anamir Cuervo is one of the people who's done the very most work on this. So it's selective protein degradation. For reasons I can't get into, Joe really wanted to look at this in slow aging mice. And so he started with the Snell-Dwarf. So the test he's doing here, lupeptin allows cells to stick in the lysosome. It blocks degradation of those proteins, so you can see which proteins move into the lysosomes. And then he isolates the lysosomes from either Snell-Dwarf liver, which is in red, or the control liver, which is in blue. And you look at the lupeptin bars and see, hey, what proteins got into the lysosomes, specifically in the Snell-Dwarf liver? And here are 3 of them, GAPDH, enolase 1, and ACADL, which is involved in cytoplasmic acetyl-CoA production. And you can see that Snell-Dwarf mice are really good at taking proteins into lysosomes. They have a lot of chaperone-mediated autophagy. Now, people know that chaperone-mediated autophagy changes with whether you've recently had a cheeseburger. So mice tend to eat a lot during the night, chaperone-mediated autophagy goes way down. The lights come on, they stop eating, and they get hungrier and hungrier and hungrier throughout the course of the day, and chaperone-mediated autophagy starts to come up. So in normal mice, not these dwarf mice, in normal mice, chaperone-mediated autophagy has a circadian rhythm. Whenever you give them food, it goes down. It's a sign of not having eaten a lot recently. But the Snell dwarf mice, and it turns out all the other slow aging mice, like the growth hormone receptor knockout mice, they think they're hungry all the time in a cellular sense. So they are leaving chaperone-mediated autophagy on, even at night, even in the day, all the time it's elevated.
And even in their hypothalamus, or what tissues do we know this for?
Hypothalamus we haven't looked at yet, but I'll show you some liver stuff. We should look at the hypothalamus. Okay, so there's the growth hormone receptor knockout mice. It's exactly the same story. He's done this now for 6 other proteins. These are the 3 I showed you. Okay, so 2 of the interesting proteins are shown here. The protein is shown on the left. One of them is CYP2A. That's a cell cycle regulator, and it is regulated by a protease that is altered in the Snell dwarf mice by chaperone-mediated autophagy. So when you're a Snell mouse, you degrade the CYP2A, and that influences, for instance, carcinogenesis, because it's important in cell cycle control. The other one, the second bar there, is c-Myc, a very famous protein. John Sedevy and his crowd showed that if you have a hemizygous mice, which has half the normal level of c-Myc, they live a long time. And all of these mice also have half the normal amount of c-Myc, and they have half the normal amount of c-Myc because it is susceptible, it's controlled by chaperone-mediated autophagy. The mRNA levels shown on the right, these are again, not transcriptional control mechanism, it's a degradation control mechanism. So this is the key part of the experiment. Joe asked, what portion of the proteins that are changed in the liver of slow aging mice, this is growth hormone receptor knockout mice, what proportion of them are moved down, and only because of chaperone-mediated autophagy? So this is a crosstab between 2 different proteomics experiments. The vertical one is lysosomal enrichment. That's the measure of chaperone-mediated autophagy. If you're over there on the right, it's because your protein has moved into the lysosomes, chaperone-mediated autophagy. That's on the right part. But the vertical axis is liver, whole protein, whole liver lysate, GERCO versus normal. If you're below the line, that means you have less of it in your liver. So the red dots there are the proteins where both things are true. There's at least a 15% decline in the liver lysate protein level, and at least a 15% increase in the accumulation of the stuff into lysosomes. So it's chaperone-mediated autophagy. You can see at a glance, there are a lot of proteins where the blue dots, where chaperone-mediated autophagy is active. If that's all you had, you'd think, wow, look at all those proteins controlled by chaperone-mediated autophagy. But most of them are not actually changed in the GERCO liver. So even though chaperone-mediated autophagy is active, it doesn't change the protein level. If you looked at the GERCO, anything below the line, you say, wow, a lot of proteins are lower in the GERCO liver, but only a chunk of them, the ones on the lower right-hand corner, are low because of chaperone-mediated autophagy. Now, are those the 91 red dots? Are there 91 proteins? Are they special, or are they just a random selection?
Uh-oh, you're gonna get to gene set enrichment. I hate to see you go this way.
No, I would never do that. Not at all. But this is the kind of gene set enrichment where a middle schooler could do it, because they're all ribosomal
Oh my God,
proteins.
okay.
And you
Um,
can do notice, right?
pardon?
These kind of like tag along with show every time.
In this case, they're highly enriched. Of
But
the 6,000 lysosomal proteins,
What's the
only
FDR?
91 were in the lower right-hand corner, and it included about 15 ribosomal proteins.
That's
That's
right.
not noise.
But I have to tease you and say, what's the FDR on this? And then you have to say it's 10 to the minus 3 million or something.
You're welcome to do that. Then the translation factors, they also surprisingly control translation. There are 2 eukaryotic initiation factors, and 2, you carry out a translation elongation factors. So the take-home message here is that of the 91 proteins that are in that lower right-hand corner, both enriched in lysosomes and decreased in Gurkha liver, they are surprisingly enriched in things that control translation or play some role in translation. Now, there were 3 others. Cytoplasmic acetyl-CoA generation just said, wow, I'll bet these mess with liver fat. And that was his next paper. That's true. So this is non-mitochondrial fatty acid synthesis. This is cytoplasmic acetyl-CoA. So the 3 proteins in red, those were red dots in that proteomics plot. The blue one, someone else had already proven was taken up into the lysosomes of Gurkha. And then the 2 green ones, ACACA and IDH1, had already been known to be CMA substrates. So all 6 of the proteins in the cytoplasm that are responsible for acetyl-CoA seem to be, at least in the liver, tightly regulated by selective degradation. Now, I'll introduce one new mouse model here, P10, a very famous gene. It's a human tumor suppressor gene. Most tumors, in order to do well and kill you, have got to have a lot of P10. It's a tumor suppressor, so they've got to knock out P10. Moving P10 down is very good for tumors, very bad for patients. Now, it's a phosphatase, and it regulates both lipids and proteins. It has a lipid phosphatase function, which is based upon phosphatidylinositol 3-phosphate, and a protein phosphatase function. It's a dual function phosphatase, and it regulates insulin signals. So it turns out others had shown that P10 overexpressor mice, they live a long time, they're lean, and they have less glycolysis, a lot of oxidative phosphorylation. And so Joe said, I wonder if this is another chaperone-mediated autophagy story, and the answer is yes. So there are 2 things to know, and they're reciprocal. P10 actually turns on chaperone-mediated autophagy. These long-lived mice, which overexpress P10, they've got a lot of chaperone-mediated autophagy. There are lovely pictures there. The little puncta, the little dots, are bits and pieces where proteins are getting, indicator colored proteins are being moved into the lysosomes. The red dots, that's a kinase dead P10. It doesn't have the lipid phosphatase, but it does have the protein phosphatase. It's only the lipid phosphatase that's dead. And you see, that doesn't turn on CMA, because that's the inference here is it's the lipid phosphatase that makes the difference. The green dots, C124S, that's a different mutant. That kills both the lipid phosphatase and the protein phosphatase. And that works too, of course. Once you've killed P10, you don't get the CMA, but it's the red dots that count. The red dots show that it is not the protein phosphatase function that counts. The thing that counts is the lipid phosphatase. All this is published a few months ago, but it's published. This is AML 12 source. So this is the first part of the story. It turns out to be a feedback loop. P10 turns on CMA, and voila, CMA turns on P10. It's a positive feedback loop. So there's a drug called buparlisib, which turns on CMA, and you can see that when you add buparlisib, P10 gets driven to the lysosomes for degradation. So all you have to do to knock P10 down is turn on CMA. Presumably these 2 factors balance one another out. The cool thing from our point of view is that P10 is another long-lived mouse in which CMA is up. We suspect that CMA mediated alteration in protein concentration is kind of a good thing, and maybe happens in a lot of other slow-aging mice. I've been begging Joe, without the slightest success, to look at drug-treated mice, but he is a powerful, creative, independent, brilliant scientist who just isn't interested in my advice on this topic. He does really good work. I'm really proud of it, but I wish he'd work on aging instead. Okay, so this is the last trailer for tomorrow's talk. Hey, surprise, these P10 overexpressor mice, they elevate my favorite protein, GPLD1. So the left slide shows the liver, GPLD1. Both males and females have elevated, of the overexpressor mice have elevated GPLD1. It's not a lot. The p-value is 0.05, but it's significantly there. Each dot is a mouse. And if you look in the plasma, it's also there. Here it's a little bit of a bigger effect, maybe a 50% effect. And again, the significant, the p-value is 0.01. So P10 overexpressor mouse is now our 5th mutant that has elevated GPLD1 in both plasma and liver, and it turns out fat. Okay, so the maybe important things that I'm not showing in today's brief talk. Ana Maria Cuervo has published a paper claiming that CMA goes down with age. She's built her whole lab around this. Maybe you can get mice to live a long time by elevating CMA. Ana Maria has made mice that have elevated CMA either globally or in the liver, and she's done a lot of very famous work on that. But CMA doesn't go down with age, it turns out, and Joe's last paper proves that definitively, both in black 6 mice, in the kind of mice that Ana Maria works, and in het3 mice. We did it, we got some data from Cynthia Kenyon on a different stock of mice. It really doesn't go down with age. It's sex specific, it's at middle age, but it doesn't go down with age. So the whole issue of CMA as the thing that goes down with age and it's really bad for you, I think at least deserves re-examination. That's just been published also. And the last dot that I don't have time to show you today, we have drugs that increase CMA. They're all inhibitors of PI3 kinase. They're not the famous ones. They're a very specific subset, the class 1 PI3K inhibitors. There are broad spectrum PI3K inhibitors. They have lots of bad side effects. They're not really good to give to people. But Joe showed that it is a subset, the ones that are specifically class 1 in terms of their PI3K spectrum of which PI3 kinases they block, which are the ones that increase CMA. So we are just now talking to the ITP. They've agreed. They are going to give one of these drugs to mice, and we'll see if they live a long time. They're going to give buparlisib, I think. No, alpelisib. Okay, take-home messages for this last talk, the CMA talk. So this is a second non-transcriptional way to mold the proteome. The first, of course, was differential translation. This is differential protein degradation. Second take-home message is that slow aging mice, at least Snell, Gerkel, and P10, have cells that think they are in fasting mode. And some of the target proteins, which are controlled in mouse tissue by CMA, are famous ones, which are already really considered important in all sorts of bad diseases. c-Myc, CYP2A, a cell cycle inhibitor, and control a lot of things related to protein translation, like initiation factors, elongation factors, a lot of ribosomal proteins, acetyl-CoA, metabolizing enzymes, the ones in the cytoplasm, which regulate whether you get fatty liver disease, for instance, and P10, a famous tumor suppressor. So I will show you one last slide just for fun as a sort of a free tip. In New Orleans, they add something to a meal called lagniappe to prove that they can give you, I'll show you just one slide because I think it's terrific work. It was done by Mike Garrett in my lab. Mike knew that, Mike, come on in, there's plenty of seats. And you're just in time for the last slide. This is really cool stuff. Mike knew, someone else had shown, if you take a baby mouse and it's a female mouse, and you expose it to odors of grown-up adult female mice, development is slowed. If you want to anthropomorphize this, you can say, I'm a helpless little baby female mouse. The world is filled with aggressive adult female mice. I want to slow down. I don't want to enter that world until I'm more mature.
This is so weird.
It is very weird, isn't it?
In the wild, female mice will kill babies.
True.
It's not true.
And in the developmental story, it works in males too. A baby male mouse, its development is slowed by the urine of adult males, and it's sex specific. So Mike said, okay, maybe since development is slowed, maybe aging will be slowed. I mean, why not? That's what's shown on the left. So these are 3 groups of mice. The green dots, they got no odor, they just got saline, they got the nose dabbed with saline from day 2 until weaning, and then after weaning from weaning until day 60. So they had only 60 days where they got a little dot of saline on their nose, had no effect on their lifespan. The red dots, they got urine from male adults, and you can see it has no effect on their lifespan at all. The blue ones are the weird ones. They got a dab of adult female urine on their nose from day 2 to day 60, but then not again. So you're affecting a developmental process. From day 60 till death, they didn't get any intervention. They didn't get any female urine. The intervention was over. And yet they lived longer than the untreated or the control male urine treated mice. So something is happening when you're a female mouse. I don't know if it would work for people. When you're a female mouse, something is happening between day 2 and day 60 that sets you on a path to slow aging. And that thing can be triggered by an olfactory signal. Sorry, not day 60, day 42. That thing can be triggered by learning through a sense that you're going to be in a world where there are an awful lot of adult female mice. It slows your development, and which others have proven, and what Mike proved was that it also slows your aging. The right panel shows, alas, it doesn't work for male mice. We didn't succeed in getting the male mice to live longer by giving them a dab of male urine on their nose. There's a hint maybe in midlife, but that's just imagination.
3 here.
No, because we had, this is a segregating stock, black 6 plus something else, and the reason is that
Okay, that explains why the median longevity is so
yes,
low.
no, these are not even pet through, right? We were bringing in, I'm not showing you the data here, it's in the paper. Half these mice lacked a vomeronasal organ. There are 2 kinds of scent receptors in mice, some that are interested in food, some that are interested in social cues. We thought if we knocked out the vomeronasal organ, which does the social cues, pheromones and things like things like that, that would kill it, but it had no effect. We saw it equally well in both mutant and non-mutant mice.
Did they look and see if the sort of like the fecundity
No,
or the reproductive status?
that's a good question. Well, we just wanted to see if it would work at all. And we have to admit someone to a waiting room apparently.
Joy is in the room.
Joy isn't working.
Yeah.
I have to do it tomorrow.
Yeah, because I don't have a mouth.
The students are coming in early.
Oh, great. They're welcome to come and join us.
Do you have any idea what pathways this is working through?
No.
Slow down.
I'd love to know that.
Yeah.
Those are all good questions.
Like, if you have any.
Tomorrow's talk is about the pathways that one would test if one wanted to test that. Yeah, that's what tomorrow's talk is about. So this is the final slide. I've shown you Joe's work and Gonzalo's work. Gina's work is the stuff that goes tomorrow. Mike Garrett did the smell thing at the very end, the last slide. The genotyping is done by Melissa and Bill. Lori, Jacob, and the new guys there, Micah, Robert, Trevor, and Lindsay take care of this immense, complicated, extremely expensive colony. If there's a young mouse that dies, you've lost $50. Each of these old mice is $500 to $600 by the time they're old enough to do something with, because you paid all that per diem. These are the students, undergraduates, mostly graduate students, also who worked on these projects. I'm really happy to collaborate with my ITP collaborators, Dave Harrison and Randy Strong. And we're really thankful to Manuel Serrano and his former student Danielle Herranz for the gift of the P10 mice. David Lombard was a coach in the background for all this stuff. So the work was supported by NIA and the Glenn Foundation.
Yeah, so I have one standard question, small one. So first one, speaking about the how we have some anti-aging like pharmacological agents that extend life in males but not in females. So there is this notion that the supposedly accelerated aging models probably are not really accelerated aging. When you it's just it's like progeria, right?
Yeah, I know
it's
about
just broken, something's broken and when you fix it, it's could we the males, we know that if it's in mammals, they males tend to live less than females. And there is like some biological component to it. To this, we're sure that these anti-aging drugs that work basically in males only are actually slowing down aging, or do they just, fix male phenotype that is broken as opposed to females?
So there are 3 or 4 interesting things there. First of all, the accelerated aging models, that's a triumph of marketing. They have nothing to do with aging. They have something to do with convincing an editor that this is really super cool. Your sick little mouse can be called a slow aging model. We never work on them. I write editorials making fun of them, but they're not germane to this issue. The idea that females live longer than males, which is certainly true for people, is true for many but not all species. And within mice, it's true for many but not all strains. And it's also, there's what we call a sex times environment interaction here. If they're virgins, you haven't had 3 mice live, the females live longer than males. But if they're non-virgins, the females live shorter than males. Because getting pregnant, if you're a female, shortens your lifespan, and you live shorter than males. And there's a batch of inbred stocks and F1 hybrids where the parent sex specificity is different. So the notion, what happens, are drugs that are male specific for lifespan, are they simply reestablishing the standard good healthy female pattern? And that's a really viable question. That's why we looked at 17 alpha estradiol. We expected 17 alpha estradiol, which is a non-feminizing estrogen, famous estrogen 17 beta estradiol. 17 alpha estradiol does not trigger the estrogen receptor, or at least it's tenfold less good. We expected it would bring our males up to the point where the females are, but it doesn't. It brings them well past where the females are. We also did pathology. For instance, the most recent pathology paper is on canagliflozin. It's an SGLT2 inhibitor, and it helps prevent glucose spikes after your donut breakfast. It has no effect on female lifespan and a great effect on male lifespan. So we looked then at pathology. We took a lot of mice that had been euthanized at 22 months of age, and we found that canagliflozin blocked 6 different kinds of age-associated change in 6 different organs, and did not do so in the females. It was male-specific for blocking the pathology. So the very plausible, very reasonable idea that we were simply correcting something in the males that the females could correct for themselves, the evidence so far is not in support of that. There are all sorts of male-specific changes in the proteome and female-specific changes in the proteome, and transcriptome changes, et cetera, et cetera. So, just 1 second. So, why these are male and female specific is complex. We have some data I'll show tomorrow, but it's not answered.